I have done general bacteriology with you, and now I'm moving on to special bacteriology, where I'll be discussing in detail each group of bacteria on the basis of their morphological features, their clinical manifestation, their pathogenesis, lab diagnosis, and whatever enzymes or toxins which they are producing leading to disease production. So, the first group that I'm going to discuss today are the gram positive bacteria. Now, there are two genera of gram positive bacteria which are of medical importance: Staphylococci and Streptococci. Staphylococci were first seen by Cox in 1878 and cultivated in liquid media by Pasteur in 1880. Both Staphylococci and Streptococci are gram-positive cocci, but they need to be distinguished on two major criteria. In fact, they have been distinguished. Number one is microscopy. Staphylococci are grape-like clusters, whereas streptococci are present in chains. Biochemically, staphylococci are going to produce catalase, that is, they are going to degrade hydrogen peroxide, whereas streptococci do not do so. So, if you look at this slide, it is an identification scheme showing you the gram positive cocci. So if they are catalase positive, they are going to be staphylococci, and if they are catalase negative, they would be streptococci. Second is the microscopy. If on gram staining you see these grape-like clusters, they are staphylococci, but if you see these gram positive chains, they are streptococci. So coming back to staphylococci, which are catalase positive and they are present in clusters, they have further been identified or differentiated into 32 different types or species on the basis of coagulase test. So if staphylococci are coagulase positive, they are Staph aureus, and if they are coagulase negative, then they are coagulase negative staph, which could be epidermidis or cephrophyticus, etc. Aureus and the coagulase negative staph have further been differentiated on the basis of beta hemolysis, mannitol fermentation test, and the yellow colored colonies which are produced by Staph aureus. Staph aureus are beta hemolytic, they are going to ferment mannitol and they are yellow colored, whereas the non-streptocoagulase negative staph, they are white in color, they do not ferment mannitol, and normally they are non-hemolytic. We would be discussing everything in detail as we move on, but this slide is very important because you have to keep this in mind as we move on. This picture is just showing you the arrangement of Staph aureus. On one side, you can see a bunch of grapes, which are black grapes, so purplish in color. On the other side, you see the gram stained slide of Staph aureus, which is showing you grape-like clusters. They are also just like grapes, and that is why it's taken from a Greek word. Staphylo means bunch of grapes, and cocci means grains or berries. Now, at the end of this topic, all of you should be able to give the morphological features of Staph aureus. You should be able to enlist the antigens, cell wall components, modes of transmission. You should be able to give describe the enzymes and the toxins and their effects. You should be able to explain the various clinical manifestations or the diseases produced by this pathogen, and you should be able to identify this in the laboratory. As I said, there are 32 species of Staph aureus. Important species are Staph aureus, and then we have Staph epidermidis and Cephrophyticus. Staph aureus is going to produce toxic mediated diseases, and then it also produces pyrogenic diseases, whereas epidermidis and Cephrophyticus are also of medical importance, as epidermidis is basically going to produce infections which are hospital acquired, and Cephrophyticus is going to produce community acquired infection and is now considered one of the very important urinary tract infections causing pathogens. Coming to its morphology or its habitat, this is very important skin pathogen, not a pathogen, a commensal. Humans are the reservoir for Staphylococci. The nose is the main site of colonization of Staph aureus, and 30% of the people are colonized at any time. The skin, especially that of hospital personnel and patients, is another very common site of Staph aureus colonization. However, it is very commonly seen in the upper respiratory tract or in the anterior nares, and it is also found in the vagina of 5% of the females, and that is what predisposes these females to toxic shock syndrome. Now, this is a question which you generally see in the exams. An SEQ is giving the scenario from a person who has a post wound infection, and you are asked to enlist 3 enzymes and 3 toxins which are produced by Staph aureus and their role in disease production. And at the same time, the examiner wants you to give the pathogenesis and any one of the toxic mediated diseases caused by Staph aureus, and how can you diagnose such a case in the laboratory. Coming to its morphology, Staph aureus, as I said, they are gram-positive cocci, which are grape-like clusters, and they are non-motile, non-sporing, and they are facultative anaerobes. Facultative anaerobes means what? That they can grow both in the presence and absence of oxygen. Now, Staph aureus produces a carotenoid pigment which is known as staphylococci, and this is going to give a golden color to the colonies. This pigment basically enhances the pathogenicity of the organisms by inactivating the microbicidal effect of superoxide and other reactive oxygen species within the neutrophils. And it's a highly resistant organism. It is resistant to not only disinfectants like phenol, it is also capable of growing in high concentration of salts, like it can grow up to an environment which have 7.5 to 10% sodium chloride. Now, this organism is very important because more than 90% of Staph aureus contains plasmids which are going to encode for beta-lactamase enzymes, making them resistant to most of the beta-lactam drugs. However, about 20% of these strains are also resistant to beta-lactamase resistant penicillins like methicillin and nefcillin. And these organisms are known as MRSA, which is methicillin resistant Staph aureus, mostly hospital acquired, or it could be NRSA, which is nefcillin resistant Staph aureus. You should know these terminologies. We will be discussing this in detail later on. Then it is MAC-A genes which is present in these organisms, and they are going to encode for the altered penicillin binding proteins, making them resistant to the methicillin resistant penicillins. Very rare strains can become either intermediately resistant or totally resistant to vancomycins, and they are then known as Visa stain, vancomycin intermediate Staph aureus, or Virsa stain, which is vancomycin resistant Staph aureus. Coming to its virulent factors associated with the cell wall. Now, the capsule is a very thin layer, or it's a slime layer which is made up of polysaccharides. 11 serotypes have been identified, and out of which 5 and 8 types are responsible for 85% of the infections associated with Staph aureus. Then it has a very important protein which is known as protein A. This is the major protein in the cell wall. It is an important virulent factor because it is going to get attached to the Fc portion of IgG at the complement binding site and is going to prevent the activation of the complement. As a result of which, C3b is not produced and opsonization and phagocytosis is greatly reduced. Secondly, it is used in the clinical labs because it is going to get attached to IgG and it forms a cogluatinate which is basically antigen antibody complex. Thirdly, this is not produced by coagulase negative staphylococci. Then we have teichoic acid. Teichoic acid in case of Staph aureus are polymers of ribitol phosphate, whereas with epidermidis they are glycerol teichoic acid, and they are going to adhere staph to the mucosal cell. Now, the lipoteichoic acid play a very important role in induction of septic shock, by inducing cytokines such as interleukin 1 and tumor necrosing factor from macrophages, and this is responsible for the endotoxic like shock which is seen with Staph aureus. Then coming to the peptidoglycan layer, the peptidoglycan layer of Staph aureus also has endotoxic-like properties. It is going to stimulate macrophages to produce cytokines which will activate the complement and the coagulase cascade, and this basically explains the ability of Staph aureus to cause clinical findings of septic shock, although they do not produce endotoxins. Clumping factor is the bound coagulase, and it is going to give the slight coagulase test positive, which we will be discussing later on. So this slide is basically showing you the cell associated virulent factor. The outermost is the capsule or the slime layer, and I have told you it is going to prevent the organism from being phagocytosed. Then it has this because it is gram positive bacteria, so it has a thick peptidoglycan layer, and this is the teichoic acid, which is also known as polysaccharide A, and this is the protein A, which we have talked about, the main virulent factor of this pathogen. And then this is the clumping factor, which is responsible for the agglutination test. Besides this, there are several toxins and enzymes which are produced by this bacteria. The most important are the toxins which are responsible for different diseases associated with staph, and three clinically important exotoxins are released by this pathogen. One is enterotoxin, which is responsible for food poisoning, and the second is toxic shock syndrome toxin, which is going to cause toxic shock syndrome, and the third is exfoliatin, which is going to produce scalded skin syndrome in young children. And then there are several exotoxins which can kill leukocytes, and they are known as leukocidins. It also produces hemolysins, which are going to break down the RBCs, release iron for growth of this organism. Beside this, it produces enzymes which also play a very important role in disease production. They are going to produce coagulase, or they produce catalase, DNase, clumping factors, staphylokinase, hyaluronidase, lipase, and protease. Now this is one of your very important examination question. You need to know the function of all the toxins and the enzymes and how they are producing disease in an individual. Coming back to a capsule, we all know that it inhibits phagocytosis, and obviously it's going to facilitate the multiplication of this organism. Coagulase is going to prevent the input of leukocyte in the infected area by producing clot in the surrounding capillaries, as a result of which these leukocytes cannot go to the site of infection and cannot kill the organisms. Exfoliatin is responsible for scalded skin syndrome, and it is going to separate the different layers of epidermidis, producing the bullae, which are a very important clinical sign of this disease. Hyaluronidase is going to break down the hyaluronic acid in the tissues and is going to facilitate the spread of this infection. Leukocidin, I've already told you, is going to kill the WBCs by producing holes in their cytoplasmic membranes. Lipases will break down the fat by hydrolyzing the bonds between the glycerols and the fatty acids. Protease is going to degrade the collagen and other tissue protein. In other words, all these are facilitating the organism in moving up and or in multiplying and producing disease. Protein A is going to get attacked to the FC part of the antibiotic and is going to inhibit phagocytosis, whereas toxic shock syndrome toxin is responsible for the rash, diarrhea, and shock associated with toxic shock syndrome. Now you need to differentiate between Staph aureus and from the other coagulase negative staph, and I showed you this in my first slide as well, and here again I'm showing you this because this is something very important. All of you are asked about it in your exams, in your viva, and as part of your SCQ and MCQs. Now, Staph aureus is going to produce coagulase, it is going to ferment mannitol, and it is going to produce DNA, which is not seen in coagulase negative staph. Then novobiosin resistance is only seen with saprophyticus, and whereas aureus and epidermidis are both sensitive to novobiosin. If you look at the color of the colonies on blood agar, on McConkey, on this nutrient agar, they are going to cause Staph aureus will be producing yellow to orange colored colonies because of staphyloxanthin, whereas epidermidis and saprophyticus are mainly whitish in color. Beta hemolysis is also a characteristic feature of Staph aureus, whereas the other staphylococci, which are coagulase negative staph, they are not going to give you beta hemolysis. So with this, I'm going to end here because this part is important. Please go through this part so that when I start talking about the clinical manifestations and the lab diagnosis, you should know what I'm talking about. So I've given you 3 or 4 MCQs all related to this part, and you are likely to see this in your exams as well. Please answer all these. First, read the scenario carefully and then give the correct answer. Read the options as well, all the options before you give or take the correct option. Thank you very much. And next, we'll be discussing the clinical manifestations as well as the lab diagnosis.